I'm Chris Cowell and Director of Research at the Sydney Children's Hospital Network. And I wish to welcome you all to what will be, I think, a very exciting webinar discussing the future of CAR T-cell therapy in children. But first of all, I'd like to begin by acknowledging that we are hosting this virtual event from the traditional lands of the Gadigal and the Bidjigal people of the Eora Nation in the east and the Barramadigal people of the Darug Nation in the west of Sydney. We acknowledge the traditional custodians of the various lands on which we are all meeting today and the Aboriginal and Torres Strait Islander people participating. We pay our respects to elders past, present and emerging and celebrate the diversity of Aboriginal peoples and their ongoing cultures and connections to the lands and waters of Australia. Once again, Chris Cowell, and welcome to our webinar today on Advanced Therapeutics, the future with CAR T-cell therapy. I first of all have some housekeeping messages for all of you. There will be a Q&A panel, and I noticed that there's already at least one question on that panel, because everyone will be in mute except for the speakers. And selected questions at the end of the webinar will be submitted to the two speakers for discussion. You can also, if you wish to reinforce that, there is an uptick with a thumbs up sign that you can use. And following the two speakers, there will be time for questions from most of the audience. We are fortunate today in having two outstanding speakers, Professor Tracey O'Brien and Dr. Patrick Schlegel. Tracey O'Brien is the Director of the Kids Cancer Centre and Head of the Transplant and Cellular Therapies Program at Sydney Children's Hospital, Randwick. She leads a team of over 200 clinical and research staff dedicated to improving outcomes for children and adolescents with cancer. Tracey has authored more than 100 publications and over 3,000 career citations in the fields of child cancer, cellular therapies, and health systems research. Tracey is the current Asia, Africa, and Australasian Vice Chair of the Advisory Committee for the Centre of International Bone Marrow Transplant Research. And she's the first paediatric oncologist to be appointed to the Advisory Board of Cancer Australia in 2019 and was recently appointed as the interim chair. Dr. Patrick Schlegel is a CAR T cancer treatment expert from Tübingen, Germany, who has recently joined the Westmead Health Precinct in Sydney as part of a multi-party partnership between University of Sydney, the Children's Medical Research Institute, and Sydney Children's Hospitals Network. Dr. Schlegel is Professor and Head of Cellular Cancer Therapeutics and is now set to lead a new research program on CAR T-cell immunotherapy that aims to bring novel treatments to patients suffering from a wide range of malignancies. This program is part of an arrangement between the University of Sydney, CMRI, and the biopharmaceutical company Bioceptor, who are developing anti-cancer therapies, in particular targeted therapeutics and immuno-oncology products targeting most types of cancer. His major focus in research and in the clinic has always been cellular therapies. This includes allogeneic stem cell transplantation and especially haploidentical stem cell transplantation. The Q&A, as I said, will follow the two talks. And I'd now like to welcome Professor Tracey O'Brien, who will commence this webinar with an overview of CAR T-cell therapy. Thank you, Tracey.
That's great. Thanks very much, Chris. Thank you for the invitation, and thanks to the Kids Cancer Research Institute for organising, the Kids Research Institute, KRI, organising the webinar today. It's my pleasure to be speaking on CAR T-cell therapies. Here's what I'm going to cover in the next 10 to 15 minutes. I'll talk about what CARs are, explain how they work, share some of the pivotal data in paediatric leukaemia. I'll also share some of the local and the Australian experience experience that we have using CAR T-cell therapies to date. I'm going to cover some of the toxicities with CAR T-cell therapies and some of the limitations. And most of my talk will concentrate on hematological malignancies, and I'll touch on some of the future use in that space as well. So CAR T cells are an example of genetically modified cellular therapy, and they fall into the umbrella of immuno-oncology, the so-called fifth pillar of cancer treatment, alongside surgery, radiotherapy, chemotherapy, and targeted therapies. And CARs have caused a great deal of excitement, both in the scientific community and in the popular press over the last decade. And the image there shown on the screen of the first successful child, Emily Whitehead, who was treated at Children's Hospital Philadelphia, went viral around the world, and it captured the hearts and the minds of so many, and so many further interest, and I think really galvanized interest in this field and application as a real game-changing therapy that we hadn't seen for some time in the cancer space. Now, I'm just going to spend a bit of time talking about what CARs are. I know we've got a very broad audience today. CAR stands for Chimeric Antigen Receptor cells. And essentially, CAR constructs all vary, but they're about sticking together the antigen recognition capabilities of an antibody with the potent immune effector capabilities of a T cells. If I say that in another way, it's a way to design a bespoke T cell that can seek out and find a specific cancer cell, can latch on, it can make more of those T cells, bring in some reinforcement, hence the popular term in the press of a living drug, and it can kill those cancer cells. CAR T cells have also been used to treat infection, and there's a lot of interesting research in autoimmune disease as well. So there are two components. One's the antigen component, and the CD19 CARs are the only commercially available product at this stage. And CD19 is expressed on many B-cell leukemias and lymphomas, including the most common form of childhood leukemia, acute lymphoblastic leukemia. There are many other target antigens in development. CD22 would be another strong candidate in paediatric leukemias. There's also an intracellular component of the CAR, and this consists of the CD3 zeta signaling domain and co-stimulatory domains together. Those two are responsible for the T cell activation and survival upon the antigen binding. The most two common co-stimulatory domains used are either CD28 or 41BB, and these are seen on the bottom left-hand side and represent the second generation CARs. And they're the ones that are used in commercial current use at the moment. Now, the clinical trials ongoing in both third generation and the fourth generation CARs, which we call trucks, are also engineered to include an inducible expression of a component such as something like cytokine IL-2. So this can be released to enhance the activation of CAR T cells, but also activate the innate immune system to kill antigen negative cancer cells. Can also be packaged up with oncogenic viruses for an added punch, suicide genes so you can turn off the cells. How does it work in practice? If once we've identified a child who's suitable for a CAR T cell therapy, we collect their blood, their T cells using a process called apheresis, insert a VAS cath to collect those cells and collect it up to a machine that we have on the outpatient wards. The T cells are collected, removed, the other cells return back to the body. Those cells are sent to the laboratory where their cells are genetically modified to incorporate this CAR construct. Those T cells, then called CAR T cells or chimeric antigen receptor T cells, amplified up, and then they're transported back, and we give them back to the patient after the patient's received some chemotherapy. First approved CAR T cell in the United States, and the only approved product here in Australia, is Tisagen-Lec-Lucel. It's a Novartis product. Trade name is Kymriah, and it was first approved in the US in 2017 for young people up to the age of 25 for acute lymphoblastic leukemia that was either refractory or to current therapies or in second or greater relapse. It was also approved a year later in diffuse large large B cell. And these are the two indications that are approved for use in Australia as well. And the construct, just as I described, is a CD19 CAR, and it uses a 4-1BB co-stimulatory domain. And this was the publication, the seminal publication that was used to form the business case for the FDA approval. This is what the product actually looks like when it arrives back in the country. This is the data from that seminal, the pivotal paper. It was published a year after the FDA approval in the New England Journal of Medicine. Shannon Maud was the lead author in 2018. There were 92 patients enrolled from around the country. It was in patients up to the age of 25. One patient in Australia contributed down in Melbourne, actually infused at the Peter Mac. 75 patients were actually received the product, the infusion of the CAR T cell, and 48 remained in follow-up. These were all heavily treated patients that were refractory to commonly used arsenals of chemotherapy and or transplant. And the event-free survival you can see on the curve there, and the overall survival at 6 months was 73% and 90%. This was incredibly exciting, much better than our historic data. The endpoint of the study was trying to beat a goal of 20%. So those results were quite extraordinary when they came out. You can see that both those curves are continuing to decline. And what that means is that the median overall survival hasn't actually been reached yet in the study, despite its commercial use. This is a slide direct from Novartis, and Novartis continue to share this data with the scientific community, with results pushed out past the 22 months that was reported in the New England Journal of Medicine out to 34 months. And although there's been a drop-off in survival, you can see that the results are still much better than the endpoint that I described of 20%, which represented sort of historical control. Now, what are some of the side effects of using CAR T cell therapy? I mean, the immune system, I think we all know, can be a weapon of mass destruction. The main side effect is what's called cytokine storm or cytokine release syndrome, and this can cause damage to many organs. To mention a couple: lungs, pneumonitis, liver, endothelial damage with cytokine elevation can cause a capillary leak, can cause vasodilatory shock. And then there's the neurological system can also be affected by a similar mechanism in a syndrome which is now referred to as ICANS, or immune effector cell associated neurotoxicity. So we understand a lot of some of the pathophysiology of cytokine storm, and we've certainly, the experience in CAR has helped inform some of the treatments in things like COVID-19, which can also cause cytokine storm. And essentially there's, pathological sustained cytokine production. In the case of CAR, it's the cytokines that are released by the CAR T cell. So there's lots of interferon gamma, and there's lots of interleukin-6 that are released following infusion of a CAR T cell. And we treat, therefore, the cytokine storm with drugs like tocilizumab, an IL-6 blocker. That we also treat with steroids from time to time to help sort of control some of the downstream continued prolonged activation of the pathways causing cytokine storm. Now, in terms of the magnitude of the side effects of CAR T therapy, they're a complex therapy, requires a team effort to manage these patients. This is from the seminal paper, New England Journal of Medicine paper, which showed in that study of 75 patients, 46% had grade 3 or 4 cytokine release syndrome, and over half the patients required admission to intensive care. Now, what we've learned since those first 70 patients is a lot, and I think we're really learning in the clinics that we're just at the sort of the tip of the iceberg, and our use is very much in its infancy. And I think the collection of real-world outcome data continues to be incredibly important and informs our use in clinical practice. And there are 3 very important registries: the CIBMTR registry, the Paediatric Real-World Cellular Therapy Consortium, Consortium and the UK Consortium, who are continuing to track post-marketing outcome data for use of CAR T-cell therapies in paediatric patients. And we're learning some very valuable things from this data. First of all, we know based on MRD, 28 days after a CAR is given, who is likely to do well and who is likely to do poorly, and that's shown on the graph on the right-hand upper side. This can help to identify who perhaps should go on and have a transplant or a second therapy. In the original study, patients with CNS disease and infant leukemia were excluded, and yet they were included in the FDA and the Australian TGA licensing. And we've since learned that the use in patients with CNS disease and infant leukemia, there is actually quite good efficacy, but for example, in infant leukemia, 30% of patients will have myeloid transformation. Similarly, we're learning subgroups of acute leukemic populations, Down syndrome, high-risk cytogenetics, where outcomes have been traditionally poor, seem to do pretty well with a CAR, and that's very encouraging. More and more data is coming out about the prolonged B-cell aplasia and the impact that that has in terms of infection rates and immune reconstitutions. And then just more recently, we're starting to understand that dose does matter, and this was published just recently, a few weeks ago, at the tandem meetings, which show that the more CAR T cells you get, the better the outcome. And then the use of prior chemotherapies in the lead up to the CAR also affect outcome. Pleasingly, the real world data has shown us that the side effect profile has improved post-market. We've got more comfortable using tocilizumab, more comfortable using steroids when we need to, using those earlier in our algorithms, and consequently, the rates of severe CRS have fallen down to 16% from 48%, and likewise fallen to 27% for the severe neurotoxicity. And I think these outcomes will continue to improve as well. Concentration is now sort of looking at beyond the cytokine storm, the short-term side effects, and learning about some of the later subacute effects of CAR T-cell therapies. Living with cancer and the risk of relapse when we've still got survival curves that continue to have not matured yet. The risk of infection lifelong. Patients who are successful with CAR need to have replacement of immunoglobulin. The long-term effects of any neurocognitive impact of the cytokine storm causing ICANS syndrome, and other late effects are being looked at and data being collected. Briefly, the Australian activity, there are 3 sites open. Royal Children's Melbourne was the first to open, closely followed by Sydney Children's here in Randwick. Brisbane's just opened up and has infused their first patient. There have been 24 patients in total that have received the Kymriah infusion of CAR T-cell therapy, ages as shown there on the screen, with the usual indications of refractory or relapsed leukemia, and 10 of the patients had a previous transplant before. There have been 2 manufacture failures, and that's one of the downsides of using an autologous CAR T-cell therapy, but the efficacy you can see there is good at around 67%. A number of patients have gone on to have a transplant after their CAR therapy, and the number of CD19 negative relapses, like some of the post-marketing data is actually quite low, lower than was anticipated. So the last 2 slides here, just like to mention some of the clinical challenges with the current use of Kymriah that's going through our minds as users of this product, and that is, can we be more aggressive in treating CRS? Can we reduce some of the initial upfront side effects? You know, how do we improve the persistence of the CAR in vivo? Is there a role for adjuvant therapy? There's some data on checkpoint inhibitors and azathioprine used in combination with CAR. Try and make the CAR stick in the body for longer. What's the role of second infusion? If you lose the CAR, can you give another one? Can you give it with a checkpoint inhibitor, and that helps it stick around? And we've certainly had patients do that, and there are ongoing studies. can we predict who should go on to receive a bone marrow transplant following their CAR, and the timing of their bone marrow transplant as well? And then lastly, is there a role to bring this therapy up front before some of the other toxic therapy? And there's an ongoing international study looking at that. Now, there are a lot more CARs. It's the tip of the iceberg. CD19 is the one that's used in commercial, but there are clinical trials reported now in CD22, CD123, targets for both B and T cell CARs. There are double CARs that are being used, combinations of CD19 and CD22, and there are lots of different ways to test the efficacy of the dual CARs, whether it be co-administration or bicistronic vectors or dual expression. The role of allogeneic CARs is very exciting, and there are a number of products in clinical trial. That this is an off-the-shelf product derived from a healthy donor. And then there's combining other gene editing tools through TALENs or CRISPR that can improve the persistence or the homing or the environment. There's one recently reported CD19 CAR. It's the first allogeneic CAR results that have been reported. It was reported at the end of last year in Lancet, and this is the UCART19 trial for ALL. It involved a handful of children, 7 children, 14 adults. It was a phase 1 study, and in this particular trial, T cells from healthy donors were used. There was lentiviral transduction with the CD19 41BB, like I've described before, and it was put together with some gene editing using TALENs to disrupt TRAC, which is the T cell receptor alpha chain, with the hope to reduce the risk of graft versus host disease. And also disrupted was the CD52 gene in that construct to help to protect the CAR from one of the lymphodepleting drugs, alemtuzumab, that we use. And that trial, phase 1, certainly showed safety and showed some efficacy. So I think that's a really exciting thing, and we'll be watching this space. There are nearly 24 other CD19 constructs that are either in early preclinical or early phase trial across a plethora of big name, well-funded academic universities, and an explosion of biotech companies that are working in this very competitive space. I'll finish there. I just wanted to call out that delivery of this therapy really is a team effort. They're complicated patients. We do about 5 or 7 a year. Wanted to acknowledge my fellow members of transplant and cellular therapy teams, my co-lead, Richard Mitchell, the other transplant physicians, Karen Adams just returned and has a great deal of experience from Cincinnati in delivering CAR T-cell therapies. Enormous amount of nursing education has been done across the hospital with Bridget Smith, Laura Chapman, the collection team, Laura and Gronya, the data management, the lab teams. My colleagues in ICU, neurology and ED have been instrumental in helping to deliver this therapy as well locally. I'm going to hand over to Patrick now, who's going to continue on the journey of what we know so far.
Okay, I hope you can hear me okay. And I want to thank Tracey for introduction in general CAR T-cell experience all over the world, basically. I want to talk to you about future directions in CAR T-cell therapy. As you have recognized, CAR T-cells have raised great expectations in cancer therapy in general. I will briefly touch major challenges of current CAR T-cell therapy, which is infrastructure and costs, new strategies for new medicines that are urgently needed, and then mainly talk about the biological challenges with regard to target antigens and tumor-associated factors, for in the end to give you a flavor of novel CAR technology that can address some. You're just not sharing it. Not sharing?
this. Yep, and then click share.
Okay, which one is shared now?
This is shared already. So if you wanted to click, yep,
Okay,
should be there.
which one is shared? Can you show me?
This one. Okay, it's all good now. Sorry.
Can see it. Sorry.
Yeah, Patrick, we can see it. Just need to expand it. That's better.
So
That's good.
that's the right one.
That's the right one.
Okay, I'm sorry. I'm sorry for that. So basically, I want to talk about new strategies for new medicines, where we need the support from the authorities as well as from clinics as researchers, and talk about biological challenges with regard to target antigens and tumor-associated factors. For in the end, to give you a flavor of a novel CAR T-cell technology that can address some of the experienced obstacles in CAR T-cell therapy. And want to mention at this point that the blue notes on the bottom of the slides are key messages on the fly for you. How does this go? Okay. One of the major ongoing discussions with regard to infrastructure and costs is the dispute of centralized versus decentralized CAR T-cell production. There definitely are arguments for both strategies. However, the more flexible approach is the on-site production in the treating hospitals, which I personally prefer. The reason why I do prefer on-site production is that the whole process of manufacturing facilitates shorter timelines and may, in long-term or mid-term, reduce costs. Above all, I do encourage and recognize that and appreciate the local investments that have been done here and are being done, as in the end, this will improve the treatment in terms of accessibility, time efficiency, and costs for our patients. If we have a short glance at the past and the present, we must correct our overall perception that CAR T-cell therapy is new. It is not. However, the treatment success with CAR T-cell therapy is quite new. Recapitulating the timelines from the past, we all agree that we need to speed up the whole development to treatment pipeline in order to deliver the cellular products to our patients. Time is critical for every single patient. For CAR T-cell therapy, we need a paradigm shift in the authorization of clinical trials. That means make it easier and faster for researchers and clinicians to learn from early patient access applications and adapt clinical trial protocols accordingly. Australia is a fantastic place to be, as this has been recognized by all parties, and things come together here and will help to develop new cellular therapeutics for our patients, and not restricted to this wonderful precinct, but to all of our patients in New South Wales, Australia, and later for patients from abroad. So broaden the application to other countries. Investments into the infrastructure for on-site CAR T-cell manufacturing at the beginning will be high. However, in the mid and long term, costs will come down as CAR T-cell therapy will become more and more a pillar in the treatment of various cancers. The increase in patient numbers will come with treatment success beyond CD19 CAR T-cells. The major problem in CAR T-cell therapy beyond CD19 is to find a good target antigen. The perfect CAR antigen on the left would be expressed on all cancer entities. It would be expressed on all cancer cells. It would be stable and high expressed on all cancer cells, and it would be homogeneously expressed on all cancer cells with no immune evasion mechanism. The reality, however, looks different. The CAR T-cell antigens are expressed also on healthy cells. Variable expression is found. Fraction of cancers only do express the target antigens, and fraction of cancer cells only are positive within a cancer entity. So, and there further is an evolutionary process that leads to antigen loss in these cancers. On the left side, the main problems with regard to CAR T-cell therapy are listed. These are the level of expression of the cancer-associated antigen, the toxicities of acute and chronic toxicities Tracey talked about, and immune evasion. The solutions are presented on the right side. So we need to do a good job in identifying new target antigens and do combinatorial targeting in order to distribute toxicities, and do multiple synchronic targeting in order to facilitate to target cancer as a whole. That means on the left side, you can see a patient with an optimal CAR T-cell target. However, the real life looks more like on the right side, in non-B lineage associated malignancies, that you would need several CAR T-cells that target different cancer antigens at the same time. And this would be true just as simple in basically all cancers that are known, despite B lineage associated cancers like AML and solid cancers, and more than one target antigen will be necessary to cure our patients from cancer with CAR T cells. I have chosen for you clinically used antibodies or target antigens, and want to point out the major obstacles in detail. That is CD19, the commonly used target antigen in CAR T cells, CD33 in AML, GD2, and ERB2. Let's start with CD19. CD19, as you can see, is more or less only expressed on B lineage derived cells. And as we are talking about targeted therapies with CAR T cells that are, in case of CD19 CARs, targeted to CD19 positive cells, CD19 CARs will target all CD19 positive cells and also eradicate B cells. That is very simple slide of hematopoiesis, where you can see the maturation of the cells, and on the right side you see the B lineage derived cells that are CD19 positive. And by targeting CD19, not only the cancer cells are targeted and eliminated, but also all healthy CD19 positive cells. This is not a problem in B cells, because B cells can, or B cell function can be substituted. B cells produce immunoglobulins, and we can substitute immunoglobulins by immunoglobulin substitution from healthy donors. However, in other target antigens, this is not true, because targeting vitally essential tissue would lead to severe damage in these tissues, such as brain, heart tissue, lung tissue, and hematopoiesis. Coming to the next target antigen, CD33, which is one of the most prominent AML-associated markers. With regard to expression on other tissues than cancer, it becomes already critical and complicated. Even though it is a clean target antigen, clean means we know exactly where the target antigen is expressed. It is critical because CD33 leads to severe immune ablation. So all the tissues that do express CD33 again would be targeted by CD33 CARs, and this would translate to immune ablation of the myeloid compartment of the immune system. And in the end, this would translate into immunodeficiency. And the time of the treatment with CD33 CAR T cells must therefore be limited, as we need this immune compartment, especially the neutrophils, to cope and to secure us from infections. That means basically all infections that require neutrophils, that is bacterial infections and fungal infections. GD2 has been used in pediatric cancer widely with the antibody dinutuximab. It is not a protein, but it's a glycosphingolipid. It causes severe pain as it is co-expressed on peripheral neural tissue, as well as the cerebellum, and causes indirect toxicity due to the inflammation. Inflammation can cause SIRS, which is a life-threatening event. In general, GD2 can be targeted in various cancers of the neuroectodermal origin, which is neuroblastoma, breast cancer, melanoma. Coming to a broadly cancer-associated target antigen is ERB2, which is well known for the targeting in breast cancer. And this becomes more complicated. As you can see, ERB2 is co-expressed in various tissues. And coming back to the function of CAR T cells will be redirected by the CAR to all these different tissues that do express ERB2, and only the overexpression and the balance of the CAR T cell function will lead to a cancer-directed therapy. However, the potency of antibodies is incomparable to CAR T cell potency, and the same is unfortunately true for the toxicities. The key message is that we need to keep identifying the best suitable target antigens and make use of combinatorial strategies in order to distribute the toxicities. In comparison to conventional chemotherapy and irradiation, the toxicity will always be very specific and not and only affect antigen positive tissue, which is not true for chemotherapy and is not true to irradiation. So the whole body will be affected by chemotherapy, and the whole body will be affected in the area where irradiation has been applied. So we should continue our work in identifying the best target antigens for our patients. But more obstacles have to be taken. There are other tumor-associated factors that impact on CAR T cell function, that are the hazardous environment with low pH, low oxygen, and low nutrient in the tumor niche, as well as soluble factors and ligands that shut down CAR T cell. For the discovery of so-called immune checkpoints, James Ellison and Tasuku Honyo have been awarded the Nobel Prize in 2018. We now make use of all these discoveries, great discoveries, to improve our CAR T cell therapies and prevent the immune checkpoint mediated shutdown in CAR T cells as well. One potential solution to address some of the challenges I talked about are adaptive CAR T cells. This is indirect CAR T cell split CAR designs. In contrast to direct CAR T cells, as explained by Tracey, CAR T are targeted to biotin or attack on adapter molecules that redirect the CARs to the tumor cells. So the adapter molecule has an antigen binding moiety and a target CAR binding moiety. The antigen binding moiety will target to the cancer-associated antigen, and the CAR is redirected to the adapter molecule via the tag on the adapter molecule. In the end, the adaptive CAR T cells are very flexible and mediate the very same effector functions such as direct CAR T cells, which are cytokine secretion, cytolysis, and proliferation. In order to give you a better understanding of the EdCAR technology, let me please share this slide with you, where we used bone metastasis of various cancers. Here it was a breast cancer. We generated a cell line and checked for antigen expression and function. In blue, you can see an antigen expression from 0%, that means no cancer cell was positive for this antigen, and 100%, which is all the cancer antigens were positive for this antigen. And in green, you can see the specific lysis. That means the activity of the EdCARs targeted to all these different cancer-associated antigens. The idea is that you screen the patient in the end for potential antigens on the tumor and perform a patient individualized treatment with one single CAR product for various cancers. That means you have one cancer, one CAR T cell for different cancers. And further, different adapter molecules can be used to engage the CAR T cells. That is full-size antibodies and fragments thereof, as well as single domain antibodies and immunocytokines to boost the effector functions. That means they do express cytokines such as IL-2, IL-15, IL-7, IL-12, IL-18 to recruit accessory cells and help to get the job done. Here the idea is illustrated. You have one CAR, you open the fridge after you have profiled the cancer of your patient, and then you take out the vials with the adapter molecules that would fit your patient. You can try to treat the patient on a patient individual level with one CAR and several different binders for this CAR. The EdCAR technology facilitates controllability of the CAR. That means you can switch the CAR T cell on by application of the antibody, and you can turn off the CAR T cells by termination of the infusion of the antibody or the antibody fragment, the binding molecule. There's flexibility in targeting. Once the patient has suffered severe toxicity, you can switch the target antigen to another target antigen in order to let this compartment recover from the toxicities. And you can combine the different adapter molecules to redirect the CAR T cells in different ways. In the end, one big advantage of the EdCAR technology is further that the EdCARs themselves do not have an intrinsic activity. So there is no limitation in cell number that can be applied to the patient. With regard to CD19 CAR T cells or other targeted therapies, it is critical to limit the number to a certain number, as lethal events have been reported if the cell number was too high, like has been reported by Steve Rosenberg 10 years ago, targeting ERB2. And in the end, what we want is to design a whole panel, a broad panel of different CARs with the ability to target different antigens and use the biotin bridge or the linker label epitope bridge to target different cancers with just one CAR T cell product. In the end, I want to point out that adapter CAR T cells facilitate to address safety and diverse targeting in one go. As a control, CAR therapy would lead, in my understanding, to great acceptance in the hospitals, because even though the CAR T cells have been released into the body of the patient, the treating physician and the whole team, including the nurses and so forth, are still in charge of the treatment. Once there's toxicity or severe toxicity occurs due to other complications like severe infections, the CAR T cell function can be terminated by stopping the infusion of the adapter molecule. And this, I hope, will lead to great acceptance in the hospitals, on the clinician side, physician side. The beauty of the system is that we can build on FDA-approved antibodies that we know and we have great experience, like with rituximab, that was the second FDA-approved antibody. We can build on the experience of this antibody, of the kinetics and the profile of the antibody. That means side effect profile, which makes it much easier to address the complications. And in the end, I want to point out one last time that we would address all these questions with one CAR targeted to all these different cancer-associated antigens and have a very flexible system. With this, I'd like to close and hand back to Chris, and I hope you could hear me okay.
Thank you very much, Patrick and Tracey, two really clear and stimulating presentations. And we'll now move to the Q&A. I notice there are a few questions, but please offer up a few questions, because Patrick and Tracey are very ready and willing for the questions. And I'd like to introduce Craig Munns, Professor Craig Munns, endocrinologist and Advanced Therapeutics Lead at Westmead, and Dr. Laura Fawcett, Respiratory Consultant Advanced Therapeutics Lead at Randwick, who are going to facilitate the questions.
Thanks, Chris. I think we'll start with a question for Tracey. And as we let these first few come through, can you tell us a bit about what was involved in the rollout of actually getting these, getting CAR T cell therapy to the patients? What services do we have to put in place at the hospital?
Oh, sure, Laura. Look, firstly, wouldn't have happened without great support by federal and state governments, so I need to stay that up front. In terms of the teams on the ground, massive team effort. This is really a therapy that requires, cross-disciplinary approach, education of all of the teams, ICU, neurology, oncology, of ourselves, pharmacy. So it was quite extensive in terms of the education. The logistics in terms of the chain of custody and the accreditation of that is quite significant as well in terms of TGA and accreditation by Novartis. So it was an enormous body of work, and I think important to say that the other sort of side of that was to get set up delivery to patients. They're still relatively rare, this group of patients that we're using CARs in the moment. I'd anticipate we'll do about 7 patients, so we certainly are learning from each one of those, and I think concentration of expertise in these sorts of niche areas and is an incredibly important thing for patient care.
Thank you, Tracey. The next question is for Patrick, comes from Richard Mitchell, who first wanted to thank you for a very interesting talk, Patrick. There are a few components to this question, but what do you think is the best strategy for local development moving forward? Should we be focusing on AdCAR-T model?
Yeah, so I'm a little bit biased because I've been working with AdCAR-Ts over the last couple of years, and I'm really a big fan of this technology. I'm very open to other technologies as well, and I'm well aware that there are different adaptive CAR technologies, and we will find out over time which is the most suitable to cure our patients. With the AdCAR technology, I think the big advantage is, again, to point out that we can address safety as well as multiple synchronic targeting at the same time, which is critical for most of the target antigens that we come across. We know CD19 CAR targeting, and before there were CD19 CARs, we had the B-specific T-cell engager blinatumomab, and we are quite aware of the toxicities and that, but proceeding to other target antigens will always require an exit strategy because we don't know what happens if, and we've had some examples where we really would have liked an exit strategy, and the AdCAR technology would facilitate this safety aspect in terms of you start the infusion. If you come across unexpected toxicities, you stop the infusion of your bind of your adapter molecule. That can be a Fab fragment with a pharmacokinetic profile that would be cleared within several hours out of the body. So the complete CAR function would be shut down and would be shut down by the treating physician, which is a very important aspect from a clinician point of view. So the safety aspect is very important for targeting other antigens than B-cell lineage-associated markers. If direct CAR T cells in the end turn out to be more efficacious than AdCAR-T technology, that is to find out in clinical trials, but to start clinical trials with indirect split CAR design is definitely what I would suggest in terms of patient safety.
Thanks, Patrick. Tracey, one question that was asked quite early in the talk from Tuba was about co-treatments with chemotherapies and CAR T-cell therapy. So she's asking about combination approaches and any cases where a combination of chemotherapy and CAR might be required.
Well, chemotherapy is still required along the way. When you collect the cells, it takes about a month to get those cells ready to infuse back into the patient. So often we need to give a little bit of chemotherapy to control disease. We also, not dissimilar to a bone marrow transplant, give some lymphodepleting chemotherapy prior to infusion of the CAR product as well to stop rejection or promote persistence of the CAR. There is some data about use of drugs to heighten the effect of the CAR, if you like. It's all in clinical trial. Dasatinib, there's certainly some data about checkpoint inhibitors in terms of being able to promote persistence of the CAR. But you don't want to kill the CAR off as well. So given chemotherapy, you need to be careful how that's done. So there is some data on that. I think we'll learn more as we go, and there's some ongoing clinical trials looking at that.
Thanks, Tracey.
Thanks, Tracey. A question for either you or Patrick. Has there been any research looking at targeting intracellular antigens or oncogenes?
Yeah, so that's a very good question because CAR T cells, as been excellently explained by Tracey, they can only target surface expressed markers, and for targeting of processed proteins that are expressed on MHC, it is much more challenging, and the expression level of these markers, the process is just very complex, and there can be some immune evasion mechanisms that drive the cancer cells into very low expression of MHC, so it makes it very difficult to target these antigens. However, what we have managed is to utilize so-called TCR-like monoclonal antibodies that are targeted to MHC. And if you look at a TCR or a monoclonal antibody, it's a very similar system. And you can target peptides that are expressed on the cell surface that are intracellular processed proteins with CAR T cells utilizing another version of either a CAR construct or the EdCAR technology.
And Craig, just to add to that, not quite on your question, but also of interest, there has been use in combining CARs with oncolytic viruses as well, which, oncolytic virus therapy is another sort of new and emerging field where you use a genetically modified virus that can selectively replicate only in the cancer cell, and you can package that up with the CAR, and there's FDA approval just recently for TVEC, it's called, and it's a modified herpes simplex type 1 virus that expresses GM-CSF, and that's packaged up into a CAR, and it's used in advanced melanoma with checkpoint inhibitor. So it's another way to sort of get double bang for your buck there.
Thank you.
Thanks both. Are there any other areas that the CAR T cells might be adapted to outside of the cancer area?
Yeah, I think there's some a plethora of areas where it's been investigated, and I think we'll see more and more of this over the years. The autoimmune disease space, I think, is really fascinating. CARs are being used there as what's referred to as bait, if you like. The University of Pennsylvania has a chimeric antigen receptor, so a KAAR CAR. So T cells that have been specifically targeting B cells that are responsible for the autoimmune disease. So the one they've got furthest away along the line is in myasthenia gravis, and that's a disease that's caused by sort of rogue antibodies against muscle-specific kinase. I'm not a neurologist, but I think they refer to it as a MuSK. And the CARs have shown some ability in the lab to direct T cells to destroy just the B cells that have that autoantibody, and that's gone into clinical trial. There's some other work being done in mucosal pemphigus vulgaris that's looking quite encouraging. There's a number of animal models in Crohn's, and then I think there's some really interesting work in MS as well. The other one that I think could have great application, Sangamo Therapeutics have this in their pipeline, and they've got a CAR that's it's actually a regulatory T cell CAR, and it's been designed to try and achieve local versus whole body immune suppression to prevent graft rejection in organ transplants. And I think that's
Oh, I think we lost Tracey there, I think.
Yeah, I may just comment on this as well. Again, coming back to the EdCAR technology, when you move away from cancer, you definitely want a controllable system. That means you want a system, if you have an autoimmune disease, that you can address whenever you need it, because just having a look at Crohn's disease is not present all the time. So you have the opportunity with an indirect CAR system like the EdCAR to treat in cycles. Once there's more activity, you redirect the CARs to the tissue, and I wouldn't talk about immunosuppression in general, but I would go further and say we can do immunomodulation, which is not necessarily immunosuppression. And I just want to comment on one thing that is very important to me personally, is that I have a very clear picture of CAR T-cell therapy in the future. And CAR T-cell therapy must be accessible for all of our patients that are in need of this treatment. And the applications will broaden, and we need to increase the infrastructure, and we need to increase the experience in this field, and not only put it in a very centralized manner. I would not find this even applicable for a country like Australia, where you have 25 million people. Many more people over time will be in need of this treatment. So you need a broad acceptance of this treatment first, and then you need to broaden the application, and you need the infrastructure in all the different hospitals to be able to cope with CAR T-cell therapy as they do with chemotherapy and irradiation therapy.
Thank you, Patrick. Sort of moving in line with that, how long would it take now if we had manufacturing on site to be able to develop CAR T-cell therapy for a specific patient?
On site here, or
Patrick
manufacturing facility?
is talking about developing it locally instead of having to send it away.
Are we talking about the procedure of having the cells in the lab and the production scheme? Yeah, so this is quite fast, and I can talk from my experience in Tübingen, where we have produced on-site CARs and cured patients with our own made CARs from our own lab. It was not these were not commercial available products, but it was our own constructs. And this is possible within 2 weeks time. And I think I have to emphasize again, time matters for our patients. Sending cells, cryopreserved cells to another country, thaw the cells, generate the product, cryopreserve the cells again, send them back. No one can tell me that this does not harm the cells. So on-site production has not only the advantage that we are much faster and we are on time, but the quality of the cells will be much better. And if we need to postpone the treatment, we can still freeze the cells down, and they have only gone through one freezing procedure then. So if we are ready to go and we have everything in the lab, we have everything set up, we would be able to generate a product right away within 2 weeks.
Craig, could I just put a clarifying statement on that for the commercial products? They are now made in Australia, not overseas. Peter Mac has just come online as the provider of the Southern Hemisphere. And there's been a great deal of research done to look at the quality of cells. So I think patients should feel very confident that there's no quality is lost because it's sent either overseas, where we do for clinical trials, or locally within Australia. I agree with Patrick that there are slicker, faster productions. There are a number of protocols around rapid production over the course of 7 and 10 days, and particularly if you're talking about giving second and third CARs, then that's also a very tantalising option for the future.
Thank you.
Just one comment on that. I totally agree that this is true for CD19 CAR T-cell therapy, but we don't know if this translates to all the other cancers. And we do know that in CD19 CAR T-cell therapy, cell number does not really matter. If you have a patient with massive tumor load, like several kilograms of tumor, you can cure the patient with 1 gram of CAR T-cells or less. And this is remarkable, but it does not translate into solid cancer. And cell number and quality of the cells will matter more and more if you approach solid cancer. So coming back to that point, in the end, we want the best product for our patients, and we have no chance to compromise in the quality of our products for our patients.
Thank you very much, Patrick and Tracey. I think it was a really good question to end up on. And it deals with a really important issue going forward around quality, efficacy, speed. And I think we're very lucky, as Tracey outlined, that we've now got our production in Victoria and at Peter Mac. And obviously we've got look forward to a future where we can have a local production as well in Sydney. But this is a national audience, so that's very exciting. So I wish to thank Tracey and Patrick for what was really quite a stimulating webinar today, and also very much the participants. It's great to see the questions that came through really at the end of the talks, and for the Q&A, it was really quite stimulating. Thank you very much to those who work in the background to pull this together, who are not really on screen, and thank you to the co-chairs, Laura and Craig, for organising this. Advanced Therapeutics is rapidly evolving, and we look forward to seeing you again. Our next webinar will be in 2 months' time, so please stay tuned. Following this webinar was filmed, and so it will be, the link to it will be emailed to you in a few days' time, and it will end up on the website, so it's available, these two great talks. So thank you very much, everybody, and have a wonderful afternoon and a great weekend. All the best.